Hi guys, Kenny here, and today we're going to be going over cancer. Now, this is a topic I take, pretty seriously, and something we usually spend more time on in the classroom. But this will at least give you some of the basic things I hope you'll take away from it. And if you ever feel like you have any questions that you'd like to ask, I'm more than willing to answer them. So please come in and give me a try. The picture you're looking at here is actually an electron microscope image of cancer. In this particular image, you're seeing cancer cells up in the upper right-hand corner that are actually invading other healthier tissue. This happens to be the alveoli of a lung, and so you're seeing cancer cells that have moved away from the breast, because this is actually breast cancer, into the lung tissue. Okay. We'll talk more about the terminology for this and how this happens, but essentially, this is metastasized breast cancer that's entering the lung. So let's go ahead and get started and take a look at our essential questions. Um, Make sure you get the key vocabulary, but please feel free to go ahead and touch base with me about any questions you may have regarding certain circumstances, and I'll help you as much as I can. So, number one, what is cancer and how is it caused? When we start looking at inside your body at the cellular level, what is the reason why cancer happens? Number two, how does this relate to cell differentiation? So when we start looking at cell differentiation, remember we talked about the fact that different cells have different structures, they have different shapes to go ahead and help them optimally go ahead and perform their job. So as they become differentiated into these different tasks that they perform, what does that do in terms of impacting them with respect to potential for cancer? And then lastly, what are the primary treatments for cancer? So when we start looking at cancer, it relates to other things that you're kind of more familiar with in your life, probably. So let's suppose you had a paper cut on your finger. Now, although the cut may have bled and stung a little for after a few days, it probably disappeared, and your finger wasn't any noticeably different. Now, how did that happen? So, how do you think your body repairs such an injury like a paper cut? And maybe something a little bigger, something a little longer. How does your body heal and repair this type of damage? Pause your video and think about this and see what you come up with. What did you think of? Hopefully, you started thinking about the fact that somehow the body is going to grow new cells. This ties into that idea of mitosis that we went over last class. Number two, how long do you think this repair process continues? How does the body know when to start the process? How does the body know when to stop this process? And what do you think the trigger is that causes the cells to stop the repair? I mean, this kind of relates too to just how big a wound can the body repair. It's a little more complicated than you may originally think, but what it all boils down to in many respects is that the cells are communicating between each other within themselves. There are a lot of communicating factors going back and forth that allow the cell to know what's going on around them and within them. And it's these signals that allow the cell to go ahead and decide that they need to go ahead and replicate and that they need to go ahead and stop replicating. But that's under normal circumstances. Now, let's assume we have some sort of mutation, as we may have talked about at the end of last section. What does that do? How does that change? And how can that lead to the body making mistakes with respect to cell division? Now, before we really start getting into this in more detail, I want to go ahead and tie this back into the idea of cell specialization and cell differentiation. Not all cells move through the cell cycle at the same rate. The rate of cell division depends largely on the type of cell. When you're an adult, most muscle and nerve cells do not divide once they're developed. Your muscle cells may replicate or divide if you're going ahead and stimulating them by exercising, causing little micro tears that look at developing these little healing patterns. But once you're an adult, there is little new cell division in these types of tissues. The exact opposite is true when you start looking at tissues that are reproducing constantly, things like skin, digestive tract, and they, in fact, actually will complete the entire life cycle in a few hours. Okay? So, when we start talking about cancer, these two different habits in terms of how fast they go through the cell cycle impacts how quickly the cancer grows and spreads. Tissues that were already fast cycling are going to be more likely to spread more quickly, and they're going to be more likely to grow more quickly. Those tissues that are more slow growing to begin with are going to advance more slowly and tend to grow less. Now, there have been many experiments related to this type of thing, and when they were first discovering these communicating mechanisms between the different types of cells, this is one of the experiments that they did. Cells in a petri dish will continue to grow until they come into contact with each other. So if you start something like a culture, you can see here in the upper left-hand corner, it starts as small little areas and they grow and grow until they actually go ahead and touch each other. Now, that's very much like the surface skin on your hand or something like that. Now, what happens if we go ahead and take some of those cells out? Let's say we had a glass slide that was growing underneath it, and we pull that out of the center. Now there's a big gap between the two sides of the culture and the cells that live there. What they noticed is that those edges begin to multiply. They begin to have cell division, produce more and more cells until they once again come into contact with each other. This is very much what we think happens when we start looking at injuries. When a cut in the skin or a break in the bone occurs, cells at the edges of the injury are stimulated to grow and divide rapidly to repair the area. Once they repair, the rate of growth returns to normal so that you don't end up with big bulges of cells. So, if it doesn't do that, if it just keeps going, this is a good example of cancer. And you would start building up a huge mass of cells, which we typically call a tumor. Now, they did a lot of experiments in these cells trying to extract whatever it was that was allowing the communication of when and when not to divide. And what they discovered was a protein that regulated the timing of the cell cycle in eukaryotic cells known as a cyclin. But cyclins are really just the beginning. There actually have been dozens of other proteins discovered that help regulate the cell cycle, but we typically classify them together with the cyclins in one of two groups. We have internal regulators, those that do communications within the cell, and we have external regulators, those that do communications outside the cell. So let's go ahead and put that here just to kind of remind ourselves. Let's shrink our size. All right, so we have inside the cell, okay, and let's have another one here outside the cell. If I can spell. Okay, so your internal inside, unlike your introns and exons that we were dealing with before, internal inside the cell, external outside of the cell. Okay, all right. Once they discovered the cyclins, one of the things that they did is they took the cyclins and the cytoplasm out of a dividing cell and they inserted it into an interphase cell. And as soon as they did that, the cell started to divide. Okay, so there's a trigger here in terms of these chemicals, and they've been able to go ahead and identify certain proteins in this case that are able to go ahead and trigger or stimulate the division process. Now, some of you may have seen a sped up version of mitosis in action, some sort of video. I actually have a little bit of one later on that we'll look at it for review. And one full cell cycle can actually vary between a couple of minutes, depending on the cell type, to several days. To give you an example, once again, skin epithelial cells have a rapid turnover rate in human body. This is important so they can replace the ones that are constantly being worn away. Remember, we were talking earlier in the semester about high-fiving and the fact that you're killing cells when you go ahead and do that. Okay? When you rub your hands together, when you go ahead and hold a pencil to move it, it's all wears away cells. So the body has to make more really quickly. Cells which make up organs like the eye, the brain, we hope they don't get damaged as often so they don't need to multiply once they reach adult size. And because of this, they are not being replaced often because they don't really get worn away. So, what this means is that cells from different parts of the body replicate at different speeds. This is due to how much they need the new cells. So those that are being damaged a lot, like the skin we mentioned, need to replace the old ones that have died. More protected organs, like your brain, don't need repair as often, as it doesn't get damaged as often and no longer needs to grow inside once you become an adult. So this has a much lower cell turnover, especially when compared to something like skin cells or your digestive tract. Let's take a look at a couple other examples. So organs which need to produce new cells continuously have the highest turnover. We're thinking about things like bone marrow, which is constantly replacing red blood cells, things like the testes, which is producing semen for adult men all the time. These are going to have a much higher turnover. But that also means that if you do develop cancer in these tissues, the cancer tends to grow and advance more quickly. Now, this term tumor is an interesting one, and most of you have probably heard it at some point. When you have an abnormality, as sometimes can occur in cells, especially those that reproduce at a rapid rate, it can lead to the production of abnormal growth, what we call a tumor or a little lump. Now, tumors of any size should be considered serious and you should get them checked out. And although benign tumors don't usually cause a whole lot of threat to a person's life, they can still be really inconvenient if they're not treated. Now, when we stop to think about this, okay, how many of you have ever had a tumor? Now, most of you are probably going to say no, but I think you'd be surprised to realize that most of you probably had. If you've ever had a major bruise where you get a lump or swelling, this is also a tumor. It's not a cancerous tumor, but it's still a tumor because the term is actually there to identify an abnormal swelling or growth of additional tissue or material. When we're talking about cancer, however, we're talking about cells that do not respond to the signals that regulate the growth of most cells. Those cyclins, those internal, external regulators we were talking about before, they are not being listened to by the cancer cell. And as a result, they also form masses, but these masses of cells, as we call them tumors, can do damage to the surrounding tissues. They are not benign. The term we typically use is malignant. We'll come back to this in a minute. So before we get too far, some key definitions I want to make sure that you guys have in your pocket. The first one is neoplasm. Neo, that prefix, means new. If any of you have seen The Matrix, the character that's the main character is Neo, meaning new. It's a new beginning, it's a new start. So, neoplasm or new growth is an abnormal mass of tissue, the growth of which exceeds and is uncoordinated compared to the normal tissues. So, this is one of the things that when we start thinking about cytology, we send off a biopsy to look at it. They're looking for these neoplasms, these abnormal tissue shapes, structures. And so, what they look like under the microscope is a good signal as to how the actual cell is behaving. Be careful using the word tumor because it's a very non-specific term and it actually simply means lump or swelling. It does not guarantee that it is cancer. Cancer is a malignant neoplasm or tumor. So malignant, I mentioned that word a little earlier, and what we mean by malignant means cancer containing. Okay, so a cancer containing neoplasm or a cancer containing tumor. Now, the last word here, metastasis, is probably to me one of the scariest terms in medicine. Okay. Metastasis means discontinuous spread of a malignant neoplasm to distant sites. Simplified, this means the cancer has left the tissue of origin and gone to a new place in the body. When this happens, typically is never going to be that the patient is full remission again. Okay. The cells are out there. They've typically gone through either the blood system or the lymphatic system, and it's just a matter of time before they pop up somewhere else. So to me, this is one of the scariest terms on this list. So as we continue to move forward, I want to go ahead and start comparing benign or non-cancerous tumors with malignant tissue. Okay. Now, when we start looking at benign tumors, they can still be pretty expansive. They can get pretty big, but they tend to grow slower, and their differentiation is typically still of that tissue. And so it's fairly traditional tissue. It's just more than there normally would be. Okay, you can get pretty big tumors, benign tumors. But the biggest thing that makes this far safer, even though it can get in the way and be inconvenient, but it's not deadly, is the fact that it does not metastasize. It doesn't leave the tissue of origin to go to a different place in the body. Malignant tissues, however, are infiltrative. They don't just produce a large mass of cells altogether. They actually kind of try and weave their way inside the normal tissue. Okay. They tend to grow faster than what we would see with a benign tumor. And their differentiation is atypical. So it's not normal. They don't look like traditional cells. They have abnormal shapes. They have different numbers of nuclei, or they're missing nuclei. Some of them are really small. Some of them are really large. They're not consistent in terms of their cellular shape, size, and structure. And when we start looking at these, we worry that once you get a tiny little bit that gets close to either the lymphatic or the bloodstream in particular, that you could break off a piece of this and it would spread to other parts of the body. This is metastasis. This is that scary thing I was talking to you about earlier. So if you have a weak stomach, I have a couple of pictures that are going to be coming up here in a little bit that are a little queasy maybe to look at. Okay, if you're one of those people, feel free to go ahead and kind of look away, pause the video, take a break. But I think it's important to kind of show the characteristics of these tissues, the cells, so that you get an idea of what we're talking about when we're looking at cancer. So here's the first one of those. When you take a look at the benign tissue, okay, this benign tissue looks fairly healthy. Okay, its color is not super abnormal. It doesn't have excessive blood vessels running through it. Even the smoothness of it is pretty consistent. That's very different from the malignant tissue on the right, which has an abnormal color. You see all these little lobes that are different sides. It's not very smooth. There tends to be more innervation, more blood kind of mixed in through it. This is what malignant looks like. And so this is what we have to kind of keep an eye out for in terms of tissue. The problem with this is a lot of the cancers that we worry about are internal. You can't look at them macroscopically like this and say, oh, that doesn't look good. I better get that checked out. So this is why it's important to make sure that we're doing screenings, that we're doing as much preventative care as we can, or keeping habits down and environments down that increase our risks for this, because there are genetic components, but there are also environmental components. But in almost every single one of these instances, what we're looking for is that key mutation that leads to cancer. And the thing is, we don't know exactly how many that is. And it's probably different for each person depending on their genetic background. In some people, the first mutation may cause cancer. But in other instances, it may be one, two, three, four, five, six, it may be many before it actually leads to a cancer like we're talking about. This may be months, this may be years, but your genome, your genetic makeup becomes less stable over time due to the accumulation of large numbers of mutations. So let's take a look at structure again. On the left, you've got a more normal cell structure. You notice that there's a single nucleus. You've got the nuclear membrane, nucleolus. All the cells are about the same size. They have their chromatin inside, about the same amount of cytoplasm. This is different when you go ahead and look at cancer cells. In most cases, they'll have less cytoplasm, hasn't had enough time to develop it because they're dividing faster. They tend to have extra nuclei or extra nucleolus or missing altogether a nucleus or nucleolus. And their chromatin, because it's going so fast, doesn't get time to wind up appropriately in terms of being ready for unwind and unzip that we talked about in transcription. So this next slide is going to show you some different types of abnormal cells. All of these are cancer to different degrees. When you start looking at it, you've got breast cancer, for example, in the upper right. You got bone cancer in the lower left. But you can see structures that look abnormal at the cellular level. They're not all the same size. They tend to have less cytoplasm, tend to have nuclei or multiple nuclei or missing nuclei altogether. And so this stands out as being infiltrative or in the way and at risk for metastasis. In these images here, cells in A represent healthy cell division. You go through the process just like we talked about. You go through G1, S, G2, interphase is over, then you go through mitosis, prophase, metaphase, anaphase, telophase. You finish up with cytokinesis, and you end up with 2 daughter cells, just 2, and they're of equal shape, and both have healthy looking nuclei. When you look at the cells here in B and C, they represent different stages of cancerous growth. But as you get to the end situation, the third column there in both B and C, it's not 2 cells, okay? And the cells that are there are not evenly shaped, okay? They're not nice and round. They don't have the same size. They don't have all the same DNA in them. They're probably missing something here, missing something there. These bottom 2, B and C, are characteristic of what we would be looking for in cytology for cancer cells. So let's go ahead and give an example of a cancer and look at it from X-ray, gross view. Now, when I say gross view, I don't mean, ew, gross. No, what I'm saying is that as it would look to the naked eye, and then the microscopic view, so you can kind of see what this would go ahead and look like. Okay. The example we have is colon cancer. And so again, if you get queasy easily, you might give yourself a little break, okay, or kind of look away as much as you can. That's what we're going to show here in a second. So you can see here that we actually have an area that shouldn't look the way it does in terms of the X-ray. This is what caused them to go in and actually take a look to try and see if they could find what the problem was. And when they did a resection or took out a piece of the colon, you can see just by looking at it, this is pretty angry tissue. Okay, it's not uniform. It's very dark in some sections. A lot more blood pouring through here and getting trapped, different lobe. It's not smooth. The color looks wrong. And then when you take a biopsy and look at that same thing under the microscope, you start to see some really dense areas where there's a lot of nuclear material, but very little cytoplasm. Cell shapes are abnormal. And you begin to realize that, yeah, this is showing a lot of the characteristic signs of colon cancer. Okay. Now, I want to go ahead and remind us of the different stages of cell division. And in case some of you haven't actually seen one of these animated versions or live videos of it, I want to give you a chance to kind of see how this happens in real time. So at this point, we're going to go ahead and watch a quick little video, and then we'll come back and talk a little bit about prognosis.
The cells of animals and plants spend the largest part of their lifetime, or life cycle, in a resting stage known as interphase. Interphase is the time between active periods of cell reproduction. It is the part of a cell's life cycle when it is growing, making specialized cell products, and when its DNA is being replicated or copied. DNA replication is actually the key event of cell reproduction because it is what makes it possible for the daughter cells to have the same genes as the parent cell. After DNA replication has occurred, tremendous changes
Remember, that
signal when the
happens during
cell
S
has left
phase.
interphase and has begun the amazing process of mitosis, the duplication of the nucleus. During the first stage of mitosis, known as prophase, threads of chromatin found in the nucleus of interphase cells condense into the compact structures seen here, called chromosomes. The chromosomes contain most of the cell's DNA and are where the genes are located. As prophase continues, the nuclear membrane disappears and tiny tubes called microtubules gather to create a structure called the spindle. In animal cells, the spindle takes shape around two structures called centrioles, not found in plant cells. It is important to realize that during prophase, the chromosomes are actually doubled and are made up of two identical strands called sister chromatids. The sister chromatids exist because the cell's DNA was replicated during interphase. A special structure called a centromere joins the two chromatids together. During metaphase, the second stage of mitosis, spindle microtubules attach to the centromeres of the doubled chromosomes. Metaphase ends when all the doubled chromosomes have arranged themselves on the equator of the spindle. Anaphase, the third stage of mitosis, begins when the centromeres split apart. Then, with the help of the spindle microtubules, the sister chromatids pull away from each other and become separate chromosomes which swiftly move in opposite directions. During telophase, the fourth and final stage of mitosis, the new nuclei make their appearance as nuclear membranes form around the clumps of chromosomes. As the new nuclei develop and the chromosomes change back into chromatin, cytokinesis, or cytoplasmic division, occurs when the cell membrane pinches off. At this point, cell reproduction has been accomplished, and there are now two new daughter cells, each of whom possesses the same genetic information as the parent cell. Now let us briefly review the process of cell reproduction. Immediately after a daughter cell is formed, it enters a long period called interphase, during which it grows and its DNA is replicated. Mitosis, the duplication of the nucleus, begins with prophase, when duplicated chromosomes take shape from chromatin and the spindle develops. In metaphase, the duplicated chromosomes line up on the equator of the spindle. During anaphase, the sister chromatids of the duplicated chromosomes split apart and move toward the poles of the spindle. Mitosis ends with telophase, during which the chromosomes change back into chromatin, nuclear membranes reform, and two nuclei are created. As mitosis is ending, cytokinesis, or cytoplasmic division, occurs, resulting in two new daughter cells that are both in interphase and which have exactly the same genes as the parent cell. Normally, cells that form the bodies of multicellular organisms reproduce only when they have been damaged, are worn out, or when a young organism is growing. However, cells of all sorts, lung, liver, blood, and so on, sometimes lose the ability to control how often they reproduce. Such cells are called cancer cells, and without effective medical treatment, cancer cells will just keep on reproducing, only stopping when they finally kill the organism in which they are living.
So, cancer at its very heart is a problem with the cell's ability to control the cell cycle, how quickly it goes through G1, S, G2, and M phase before dividing, and can it get all of the things it needs to get done, and then do so in a way that allows it to all occur at the appropriate time. Now, one of the activities had you looking at the relationship between the spindle and the timing of the cell cycle. Since the spindle is moving the actual chromosomes to where they need to go, it is important that they are actually at the right place at the right time to allow the cell to finish all of these divisions before it creates the new daughter cells so that they have all of the genetic material they need to have to function appropriately. So, when we start thinking about cells and cancer, now we have to start really thinking about how the cells are different and what that may mean in terms of how survivable the diagnosis of cancer is. This is what we call prognosis. What is the prediction of the outcome? There are many different criteria that are coming into play as we start looking at the different types of cancer. What is the grade of cancer or the stage? What's the histology routine criteria? How are we able to identify it and how are we able to look for it? What are the patient characteristics? You can't overlook the patient's overall health outside of the cancer. Somebody who has lived a very healthy life otherwise is going to have a better chance at fighting the cancer and surviving for more years. And then lastly, what are the different treatment considerations? How can you treat it? There are certain types of cancers, like blood cancers, for example, that you can't cut out. So surgery is not really an option for these individuals. Okay. So how late a stage is it? How early did you catch it, right? How bad are the cells? How advanced is the stage of cancer when it's discovered? What is the patient's overall health otherwise, outside of the cancer diagnosis? And what treatment options are available for that individual patient? Now, I'm probably going to have to look for a new set of statistics, but they're not always easy to find. These are from 2010, so they're already 10 years old, looking at the different types of cancer diagnoses, estimated new cases, and deaths. When we go ahead and start looking at this, you'll notice that the highest is those cancers that we often hear about for men, prostate cancer, and for women, breast cancer. Beneath that, we start looking at lung and the bronchi. A lot of that is still related to smoking or vaping and other sorts of diseases where we're inhaling things like that. Colon rectal cases, we're starting to do more screenings when we start looking at colonoscopies and things like that to try and identify cases earlier to allow us to be able to treat them. So when you start comparing these types of lists and then you look at the estimated deaths down below, uh, it is interesting to notice that when you start looking at the numbers with respect to like colon rectum cancer, prostate cancer, okay, the death rate is higher than what we would like to see. And so we're working on developing treatments and diagnoses to catch these earlier and intervene in a way that can go ahead and solve the problem. In fact, breast cancer, which used to be one of the biggest ones in female, is now in second, right? Lung cancer, again, largely related to smoking, is still the leading killer in terms of cancers, at least in 2010. Very similar results on the male side. Okay, prostate cancer, which is the highest diagnosed portion, has high death rates, but also still surpassed by lung cancers, again, primarily related to smoking. Okay. All right. So, cancer grade, what does this mean? The other term you may have heard is what they call a tumor grade. This is based on the microscopic features, what we call the cytology or histology of the sample that was set off from biopsy. Low grade cells are going to be fairly normal looking. They may have an abnormal looking cell here or there, but by and large, the cells are uniform in shape and have pretty normal looking conditions in terms of their nuclei, in terms of the number of nuclei, the amount of cytoplasm, these types of things we mentioned earlier. As you get into more moderate stages or grades, you're starting to look at a little bit less consistency in terms of the cell structure, size, amount of cytoplasm, number of nuclei. And when you get to high grade cancers or high grade tumors, it's very unpredictable. All sorts of different sizes, extra missing materials, and very infiltrative. So it's mixed in with the more healthy cells. And so that's the problem we start looking at. Which brings us back to the concept we were talking about before that I said was one of my scariest things in human medicine, and that's metastasis. Now, metastasis is the spread of the disease from one organ or part to another non adjacent organ or part. This is typically transported through the blood vessels or lymphatic system as a piece of the tumor breaks off from a primary tumor and then begins to circulate to another part of the body. This then allows it to grow in normal tissues in other places rather than where it originated. Now, most tumors can metastasize, but do so in varying degrees. There are certain cancers like basal cell carcinoma, which rarely metastasize. When the tumor cells metastasize, the new tumor is called a secondary tumor, and its cells are like the original tumor. So, that picture we showed you at the very beginning of the presentation is breast cancer cells metastasized to the lung. This is a secondary tumor, but it's made up of abnormal breast cells. They're not lung cells, so this is not lung cancer. This is metastatic breast cancer. Okay, so if we were dealing with these types of things, we got to think about where it came from, tissue of origin, and that's going to determine largely how we treat a particular type of cancer. So let's talk a little bit about treatments. Here are five of our traditional treatments that we start looking at. We're always looking for new treatments, and this may even be a little outdated at this point, as I am no longer directly involved in areas where they do this kind of work. But surgery still tends to be our number one option. If you can cut it out, then it's just gone. Okay. We start looking at chemotherapy and radiation, two commonly used treatments to try and clean up after surgery, or if we can't do surgery immediately. And then things like monoclonal antibodies, you'll see stem cell use, you'll see immunotherapy. These are all different treatments that are being worked with to try and lower the levels of cancer or to kind of reverse some of the impact of the cancer cells. The choice of treatment, however, depends on the location of the material, the tissue, the grade of the tumor, and the stage of the disease. So earlier we can catch it, the more we can feel successful with one of these different stages, especially things like surgery. So some surgeries are very effective in terms of removing cancer, especially if we're talking about early stage cancers. This is especially true for non-blood related cancers. And in some cases, they can remove the entire area, get all of the cancer cells, and then the patient is not going to have to deal with it again. However, this is not always possible, as it can be difficult to pin down exact extent of the tumor growth or the boundary to where it extends to. And many of the cancers have the ability to metastasize. And so, if any of those tumor pieces have broken off and entered into the bloodstream or entered into the lymphatic system, even if you think you cut it out, you may have missed something that got into those systems and spread to other parts of the body. Some examples of surgeries include mastectomies, breast cancer, or prostatectomy, it's prostate cancer. Now, these are becoming increasingly common for patients that have these particular types of diseases, and it's not considered a big deal much anymore. In fact, people like Angelina Jolie, for example, she took the extreme measure. Her family had a high incidence of breast cancer, and so before she got it, she actually had both breasts removed. Now, in her line of work, she just got implants, and it wasn't a big deal. That's a pretty aggressive approach. Um, my father-in-law had a prostatectomy to remove the prostate for his cancer. Hopefully, they had hoped to catch it before it left the boundary of the prostate, but were, in his case, unable to actually get all of the tissue before it had spread. Chemotherapy. Chemotherapy is the treatment of drug or ailment, in this case, cancer, with drugs. Most people, when they talk about chemotherapy, they're thinking about cancer, but it doesn't have to be relegated to that. Chemotherapy simply means therapy or treatment with chemicals. And so you probably have all had some low level form of chemotherapy just in terms of using Tylenol, Advil, antibiotics, these types of things. Okay. But when we say the term chemotherapy, we're typically referring to using cytotoxic drugs that target these rapidly developing cells. Um, Although it's not perfectly targeted towards just cancer, we're hoping to get better with some of this in terms of our ability to target particular cells as we potentially use things like CRISPR-Cas9, which we'll talk about next semester when we start looking at genetic engineering techniques. Now, some drugs work better together than alone and are often given in combination. When we use more than one form of therapy together, it's known as combination therapy. Common areas where we would use chemotherapy include things like leukemias, lymphomas, but chemotherapy is a common treatment for many, many different types of cancer. Another one you'll commonly hear about is radiation therapy. This is using irradiation or radiotherapy to go ahead and kill cancer cells or shrink tumors using somewhat dangerous ionizing radiation. Examples, again, many different cancers use this. This is common in breast cancer and prostate cancer. Sometimes the radiation therapy is used in combination, as we mentioned before, with chemotherapy, as the chemotherapy can help us target the radiation so that it's getting to the right source tissue. Using two or more types of therapy together is what's called adjunct treatment. To go ahead and say this was the primary treatment, and then we're going to use this with it. This is adjunct treatment. On the next slide, I will show you the potential benefits of especially these two particular treatments. In this particular picture, you can see on the left, there's a fairly large tumor in the breast tissue. This is pre-chemotherapy. And then on the right, you have the same individual post chemotherapy and radiation. And you can see that the tumor's size has shrunk dramatically. So, under the correct circumstances and used correctly, radiation chemotherapy can be quite effective. And then we can sometimes go ahead and use surgery at that point and reduce the risk of its spread to other tissues. Other types of tissues, including treatments, including things like immunotherapy and stem cells, are less tested, less well researched, and so we don't know if they are as effective as what we have seen in terms of chemotherapy and radiation. Immunotherapy is a large and diverse group of strategies designed to induce the patient's own immune system to fight the tumor. Some places where we've had some success in this include superficial bladder cancer, renal cell carcinomas, melanomas, these types of things. This category sometimes includes stem cell transplants, and you're seeing more and more use or exploration of stem cells in trying to treat cancer. Now, stem cells are characterized by the abilities of self renewal and potency. If you remember when we were talking about the different stages of development, those early embryonic cells are closer to what we would call totipotent, or have the ability to become any cell in the body. The longer the cells go on before they are harvested, they are more and more limited in what they can go ahead and become until they go from totipotent to pluripotent. And later stage ones, when we start looking at adult stem cells, are pretty limited in the small range of cells that they can go ahead and form. But the nice thing is we can extract them from the patient. So there's less chance of them being rejected or anything like that. Here are some examples. This is just one potential use of adult stem cells. In this particular case, you've got an intestinal stem cell, and we could go ahead and extract it and use it to manufacture either absorptive cells or goblet cells or any of the other different types of intestinal cells that are produced by this particular stem cell. However, it's never going to produce lung tissue. It's never going to produce nervous tissue. This is an adult stem cell that can only produce cells that are in that particular region, intestinal in this particular instance. So, a couple things I want to highlight. When we start understanding it, make sure you know the vocabulary. Chances are, unfortunately, really high that most of you will have somebody in your life that ends up having to go through cancer. And the best tool you can have is the knowledge of what they're talking about so that you can make the best treatment decisions for your loved family member. Okay. When we start thinking about this, know what metastasis is, know what a tumor is, know what a neoplasm is, know the difference between malignant and benign, and start thinking about all these different treatments. What can surgery do? What can go ahead and do with chemo or radiation or some combination therapy that uses multiple forms of treatment? Okay. If you have any questions, guys, please reach out. I can't say that I'll answer all of them, but I can try and help you and at least point you in the right direction. All right. I hope you guys are doing well. Take care of yourselves. I'll talk to you again soon. Take care. Bye bye.